For decades, neuroscientists assumed brain maturation followed a linear sequence of growth and passive decay. They viewed neurons as the sole architects of this process, determining circuit strength through internal mechanisms. Modern high-resolution in vivo imaging reveals a different reality. Innate immune cells patrol the brain parenchyma, physically engulfing and ripping away active dendritic spines. The tetrapartite model identifies the synapse as an intersection where neurons, microglia, astrocytes, and the extracellular matrix engage in constant biochemical dialogue. Within this framework, synaptic pruning is an active, immune-regulated process. This mechanism highlights a central biological paradox in human neurodevelopment. We do not achieve maximum cognitive capacity at our highest physical connectivity. Structural decay is a requirement for cognitive power. Biological efficiency favors the most refined network. An optimal brain achieves peak performance by eliminating supernumerary synapses until only the most essential circuits remain. During early childhood, the human brain produces a massive excess of synaptic connections. Cortical density reaches levels roughly 50% above adult norms. This dense, hyper-connected network provides the redundancy required for a child to learn. It fosters an exploratory cognitive state, optimized for flexible learning and rapid environmental sampling. This overproduction halts in early adolescence, replaced by an acceleration of targeted synaptic pruning. Glial cells selectively eliminate weak or redundant connections. The remaining circuits become heavily myelinated and stabilized, creating the physical infrastructure required for high-speed signal processing. This chart maps synaptic density against cognitive traits. Notice the exact inverse correlation between raw synaptic density and adult executive control. This efficiency comes at a cost. As density plummets and processing speed solidifies, the network loses the capacity to rapidly master novel tasks. Achieving the adult cognitive state requires an irreversible biological phase transition executed entirely by glial networks. Long-term depression can shrink a synaptic spine, but it cannot physically clear the remaining material from the tissue. Microglia and astrocytes act as the primary executioners to clear this detritus. To accomplish this, the brain repurposes innate immune pathways originally evolved to destroy invading pathogens. This diagram maps the precise receptor-ligand interactions driving microglial phagocytosis. The complement cascade utilizes C1q and C3 proteins to tag synapses. Parallel to this, TREM2 receptors bind externalized phosphatidylserine, triggering engulfment. Meanwhile, don't-eat-me markers like the CD47 SERP-alpha axis protect healthy tissue, overriding complement tags. The retention of any single memory, or the mastery of any procedural skill, relies on this continuous biochemical dialogue at the synaptic membrane. This selection process operates on an algorithmic scale. It mirrors magnitude-based dropout methods used to train artificial neural networks. Neurons utilize local data to calculate the relative importance of their connections. Applying principles similar to the Fisher information matrix, synapses conveying highly predictable, salient sensory inputs are stabilized. Those transmitting noisy, high-entropy signals are progressively weakened until they are flagged for immune elimination. This sorting requires massive metabolic energy. The brain cannot execute this dynamic immune clearance concurrently with active daytime environmental sampling. This dual-axis visualization maps cortical synaptic density against delta-wave EEG power. The data proves non-REM slow-wave sleep is the primary theater for synaptic downselection. Deep sleep partitions the workload, driving generalized pruning that results in a synced decline of physical synapse counts and delta waves. While salient episodic memory traces are shielded and transferred to the neocortex, microglia actively dismantle the vast majority of weak or unshielded connections. Chronic sleep deprivation halts microglial clearance. Without the necessary operational window, the brain remains trapped in a saturated, computationally inefficient state. Clinical models historically treated the central nervous system as an isolated vault, locked behind the blood-brain barrier. The mechanics of synaptic pruning show this is not the case. Microglial phenotypes are linked to the gut-brain axis. These cells rely on continuous metabolic byproducts from the gastrointestinal tract, specifically short-chain fatty acids, to maintain their healthy state. Microbial dysbiosis driven by poor diet, infection, or severe chronic stress halts this vital gut-brain signaling. Microglia are forced into an amoeboid reactive state, where they may ignore inhibitory signals and indiscriminately destroy functional synapses. Microscopic pruning also requires macroscopic plumbing. The glymphatic system pumps cerebrospinal fluid through deep brain tissue, acting as a mandatory waste clearance network. During sleep, perivascular channels must physically flush out the volume of molecular detritus generated by microglial pruning. If this fluid clearance system fails, cellular debris accumulates in the interstitial space. This buildup triggers localized neuroinflammation and forces an inappropriate reactivation of the complement cascade. Nanoscale cognitive sculpting is completely dependent upon the upstream stability of peripheral inflammation and fluid dynamics. Modern research identifies many severe psychiatric diseases as connectopathies. These conditions arise when the absolute magnitude or timing of glial pruning deviates from a healthy baseline. This lifespan graph plots relative synaptic density, mapping 3 major pathologies directly against a healthy baseline. Looking at the early childhood developmental window, the orange curve identifies autism spectrum disorder. Functionally, this represents a state of pathological underpruning. The molecular driver is often linked to hyperactive mTORC1 signaling. This hyperactivity suppresses neuronal autophagy, preventing the initial degradation of redundant connections. Because the synapses are never weakened, microglia never receive the signals to clear them. The brain retains a vast network of noisy, redundant infrastructure. This dense, unpruned cortical network leads to the sensory processing overload and delayed information filtering characteristic of the disorder. In late adolescence, the pink curve reveals the structural reality of schizophrenia. Contrasting sharply with autism, schizophrenia represents a state of pathological overpruning. The strongest genetic risk factor involves structural variations in the MHC locus. These variations drive severe overexpression of the complement C4a gene inside the brain. This genetic overabundance blankets healthy connections in inappropriate eat-me tags. Microglia respond to these chemical orders, aggressively stripping prefrontal dendritic spines far below baseline requirements. The characteristic onset of psychosis and executive dysfunction in early adulthood aligns with the destructive zenith of this runaway immune cascade. In late adulthood, the blue curve tracks the terminal plunge in synaptic density associated with Alzheimer's disease. As amyloid beta plaques accumulate in aging tissue, they disrupt the local microenvironment and create a highly toxic chronic inflammatory state. This sustained inflammation inappropriately unleashes the dormant C1q and C3 complement cascades, awakening the destructive mechanisms that sculpted the adolescent brain. Tricked by these chemical markers, reactive microglia systematically tag, digest, and destroy mature, functional memory synapses long before widespread neuronal cell death occurs. From infancy to senescence, the innate immune system continuously curates the structural architecture of human cognition. Future treatments will likely focus on manipulating microglial phenotypes to actively defend the connectome, moving past the limitations of simple symptom management.